Welcome to Chapter 9, Section 3, The Effects of pH on Microbial Growth. Learning objectives. Illustrate and briefly describe minimum, optimum, and maximum pH requirements for growth. Identify and describe the different categories of microbes with pH requirements for growth: acidophiles, neutrophiles, and alkalophiles. Give examples of microorganisms for each category of pH requirement. Yogurt, pickles, sauerkraut, and lime-seasoned dishes all owe their tangy taste to a high acid content. Recall that acidity is a function of the concentration of hydrogen ions, or hydronium, and is measured as pH. Environments with pH values below 7 are considered acidic, whereas those with pH values above 7 are considered basic. Extreme pH affects the structure of all macromolecules. The hydrogen bonds holding together strands of DNA break up at high pH. Lipids are hydrolyzed by an extremely basic pH. The proton motive force responsible for production of ATP in cellular respiration depends on the concentration gradient of protons across the plasma membrane. See cellular respiration. If hydrogen ions are neutralized by hydroxide ions, the concentration gradient collapses and impairs energy production. But the component most sensitive to pH in the cell is its workhorse, the protein. Moderate changes in pH modify the ionization of amino acid functional groups and disrupt hydrogen bonding, which in turn promotes changes in the folding of the molecule, promoting denaturation and destroying activity. The optimum growth pH is the most favorable pH for the growth of an organism. That's pretty straightforward. The lowest pH value that an organism can tolerate is called the minimum growth pH, and the highest pH is the maximum growth pH. These values can cover a wide range, which is important for the preservation of food and to microorganisms' survival in the stomach. For example, the optimum growth pH of Salmonella species is 7.0 to 7.5, but the minimum growth pH is closer to 4.2. Most bacteria are neutrophiles, meaning they grow optimally at a pH within 1 or 2 pH units of the neutral pH of 7. And see this figure like right here. Most of them are neutrophiles. Most familiar bacteria, like Escherichia coli, Staphylococci, and Salmonella species, are neutrophiles and do not fare well in the acidic pH of the stomach. However, there are pathogenic strains of E. coli, Salmonella typhi, and other species of intestinal pathogens that are much more resistant to stomach acid. In comparison, fungi thrive at slightly acidic pH values of 5.0 to 6.0. Microorganisms that grow optimally at pH less than 5.55 are called acidophiles. For example, the sulfur-oxidizing Sulfolobus species, isolated from sulfur mud fields and hot springs in Yellowstone National Park, are extreme acidophiles. These archaea survive at pH values of 2.5 to 3.5. Species of the archaean genus Ferroplasma live in acid mine drainage at pH values of 0 to 2.9. Lactobacillus bacteria, which are an important part of the normal microbiota of the vagina, can tolerate acidic environments at pH values of 3.5 to 6.8 and also contribute to the acidity of the vagina, pH of 4, except at the onset of menstruation, through their metabolic production of lactic acid. The vagina's acidity plays an important role in inhibiting other microbes that are less tolerant of acidity. Acidophilic microorganisms display a number of adaptations to survive in strong acidic environments. For example, proteins show increased negative surface charge that stabilize them at low pH. Pumps actively eject hydrogen ions out of the cell. The changes to the composition of membrane phospholipids probably reflect the need to maintain membrane fluidity at low pH. At the other end of the spectrum are alkalophiles, microorganisms that grow best at pH between 8.0 and 10.5. Vibrio Cholera, the pathogenic agent of cholera, grows best at the slightly basic pH of 8.0. It can survive pH values of 11.0, but is inactivated by the acid of the stomach. When it comes to survival at high pH, the bright pink Archean Natronobacterium found in the soda lakes of the African Rift Valley may hold the record at a pH of 10.5. Oh, we have a picture of them. Here we go. Extreme alkalophiles have adapted to their harsh environment through evolutionary modification of lipid and protein structure and compensatory mechanisms to maintain the proton motive force in an alkaline environment. For example, the alkalophile Bacillus firmus delivers the energy from transport reactions and motility from a sodium ion gradient rather than a proton motive force. Many enzymes from alkalophiles have a higher isoelectric point due to an increase in the number of basic amino acids than homologous enzymes from neutrophiles. Microconnections. Survival at the low pH of the stomach. Peptic ulcers, or stomach ulcers, are painful sores on the stomach lining. Until the 1980s, they were believed to be caused by spicy foods, stress, or a combination of both. Patients were typically advised to eat bland foods, take antacid medications, and avoid stress. These remedies were not particularly effective, and the condition often recurred. This all changed dramatically when the real cause of most peptic ulcers was discovered to be a slim, corkscrew-shaped bacterium, Helicobacter pylori. This organism was identified and isolated by Barry Marshall and Robin Warren, whose discovery earned them the Nobel Prize in Medicine in 2005. The ability of H. pylori to survive the low pH of the stomach would seem to suggest that it is an extreme acidophile. As it turns out, this is not the case. In fact, H. pylori is a neutrophile. So how does it survive in the stomach? Remarkably, H. pylori creates a microenvironment in which the pH is nearly neutral. It achieves this by producing large amounts of the enzyme urease, which breaks down urea to form ammonium and CO2. The ammonium ion raises the pH of the immediate environment. This metabolic capability of H. pylori is the basis of an accurate, non-invasive test for infection. The patient is given a solution of urea containing radioactively labeled carbon atoms. If H. pylori is present in the stomach, it will rapidly break down to urea, producing radioactive CO2 that can be detected in the patient's breath. Because peptic ulcers may lead to gastric cancer, patients who are determined to have H. pylori infections are treated with antibiotics. Okay, I'm going to read you a little bit from this Discover magazine article. For years, an obscure doctor hailing from Australia's hard-scrabble west coast watched in horror as ulcer patients fell so ill that many had their stomachs removed or bled until they died. That physician, an internist named Barry Marshall, was tormented because he knew there was a simple treatment for ulcers, which at the time afflicted 10% of all adults. In 1981, Marshall began working with Robin Warren, the Royal Perth Hospital pathologist who, 2 years earlier, discovered the gut could be overrun by a hardy, corkscrew-shaped bacteria called Helicobacter pylori. Biopsying ulcer patients and culturing the organism in the lab, Marshall traced not just ulcers but also stomach cancer to this gut infection. The cure, he realized, was readily available: antibiotics. But mainstream gastroenterologists were dismissive, holding on to the old idea that ulcers were caused by stress. Unable to make his case in studies with lab mice, because H. pylori affects only primates, and prohibited from experimenting on people, Marshall grew desperate. Finally, he ran an experiment on the only human patient he could ethically recruit: himself. He took some H. pylori from the gut of an ailing patient, stirred it into a broth, and drank it. As the days passed, he developed gastritis, the precursor to an ulcer. He started vomiting, his breath began to stink, he felt sick and exhausted. Back in the lab, he biopsied his own gut, culturing H. pylori and proving unequivocally that bacteria were the underlying cause of ulcers. So this satisfies Koch's postulates. Actually, he took it from the other person, and then he's the reinfection. Anyway. All right, and after that's an interview that I'm not going to go through all, but this is from Discover Magazine from March 2010. The doctor who drank infectious broth gave himself an ulcer and solved a medical mystery. That brings us to the end of Section 3. Next time is Section 4, Temperature and Microbial Growth. Until then.